I am Jacques Fantini, professor of biochemistry at the University of Aix-Marseille. And the purpose of my talk today is to start from the broad antiviral activity of Nigella sativa against a huge diversity of viruses. And the question that I am asking today is whether there is a possibility to identify a unique mechanism of action, or can we consider that many phytochemicals derived from Nigella sativa have different mechanisms of action? So on the next slide, you have a quote from a paper by Villanueva and collaborators, which is entitled Pharmacological Activities of Phytomedicines, a Challenge Horizon for Rational Knowledge. In fact, on a global perspective, what we need when, as a biochemist, I am speaking of phytotherapy, is that we need a real rational, complete, and widely accepted theory to explain phytotherapy efficacy and the mechanism of action in herbal drugs. So when we look at Nigella sativa, there is broad biological effects, anti-cancer, anti-parasites, antibacterial, and so on, anti-inflammatory. And so I will focus my talk today on the antiviral activity. What can we say about the biochemical or chemical or mechanism of action of the phytochemicals derived from Nigella sativa? We are, in the next slide, at the era of artificial intelligence. So you know, or you may know that AlphaFold is making significant progress in the structural elucidation of many proteins, but I think that it is a little bit hazardous to trust what artificial intelligence is giving to us for the following reasons. When you look at this very beautiful structure, you can see that a phytochemical derived from Nigella sativa, which is called isoquercetin, binds really finely to a protease from a virus, and in this case, dengue virus. And so you can say that, okay, the phytochemical binds to the viral proteins, so it inhibits the function of this viral protein, and this is the biochemical mechanism of action of Nigella sativa against this virus. But the situation is really more complex than that, as you can see on the next slide, because you have several compounds in Nigella sativa. This is just a selection. We have a dozen of phytochemicals. And what you can see is that every time you perform a docking experiment on a given viral protein, you will have different results. And the amino acids that constitute the ligand binding pocket of each phytochemicals on the protease of this dengue virus, you can see that the region of the binding is different for all the phytochemicals. So is it possible, is it rational to admit that in Nigella sativa you have, let's say, 20 active compounds? Each of these 20 active compounds binds to the same protease of the virus, and then even if it binds at different locations of the protein, it will inhibit its function. So I think this is not reasonable to make such conclusion, because this is an endless process. Each time you will look to a particular viral protein, you will be able to find a docking, because these phytochemicals are small molecules with aromatic compounds, with chemical groups that really like to interact with protein. So I think that this is not a rational strategy to identify a mechanism of action. On the next slide, you have one of the reasons why we should beware of artificial intelligence. I'm not working against artificial intelligence, of course. It can give really interesting information. But beware of docking. When you try the docking of a molecule on a protein, then most of the time, the initial docking is not stable. And what you can see is that if you go ahead the docking, you have the molecular dynamic simulation, and then, as it can be seen on this slide, the initial docking is not stable. So on the next slide, you can see that this is also a problem with AlphaFold. AlphaFold has resolved the structure of many proteins, but when the part of the protein is disordered, then AlphaFold cannot do anything. And instead of providing a reliable structure, then AlphaFold give you a lasso-like domain, a lasso-like folding, which is represented in this slide. And this does not give you a real information on the structure of the protein. And of course, how can you dock on this protein? So we have to add some human touch and think a little bit. And this is what this slide is showing. We need a unitary mechanism. So let's stay simple. A virus infects a cell by an interaction with the host cell membrane. So this is at the level of the virus-host interaction that we should primarily look for a mechanism of action. On the next slide, you will see that, next slide, okay, you will see that several distinct viruses have a convergent evolution to interact with the host cell with the plasma membrane. And then in the plasma membrane, not everywhere on infection platforms that are represented by lipid rafts. Lipid rafts are domain of the plasma membrane enriched in ganglioside and cholesterol. And this lipid rafts attract the virus. And what you can say is that it is the same mechanism for many, many viruses. And on the next slide, you can see that it works on HIV, it works on SARS-CoV-2 for the COVID-19 pandemic. It works also to explain why several variants of SARS-CoV-2 are able to infect the cells and to win the race from the previous variant, because the rafts are electrically negative charge domains. And the proteins of SARS-CoV-2 begins to be more and more electropositive, more cationic. So the initial strain was reasonably cationic, and then when you move to the Delta, Omicron, and the last Omicron strain, the GN1, you have now viruses which are highly, heavily cationic. And so this part of the knowledge of the host virus interaction is very interesting to test the antiviral mechanism of molecules that have a broad antiviral activity on several viruses. And this I will show you on the next slide, where I performed a structural analysis of two distinct antivirals. I will not give you the information of what these antivirals are, because this is not the purpose of my talk today. But you have two antivirals with a broad antiviral range activity. One is represented in yellow, the other is represented in green, and what you have in blue and red is the surface envelope glycoprotein of a virus. The blue areas are cationic and the red areas are anionic. What you can see is that the surface of the protein is covered by cationic zones. And then the question is, what about Nigella sativa? I think that the research has not been done, but it is really, really important to check that the phytochemicals derived from Nigella sativa, which have a broad antiviral activity, may in fact inhibit, like other molecules with broad antiviral activity, so may inhibit the interaction of the virus with lipid raft. And so you have to show that they bind to the cationic zones of the envelope proteins of the viruses, and then interfere with the binding to lipid rafts, so to the initial step of the infection. On the next slide, you can see that thymokinone, next slide, if possible. Thank you. So on this slide, you can see that thymokinone is the most active antiviral phytochemical of Nigella sativa. And as I showed you on the preceding slide, is that this phytochemical really binds to the electropositive zones of enviral glycoproteins. And so this product is able to block the binding of many, many viruses, HIV and other viruses that are represented here. So I think that now we have really a unique mechanism of action that is really rational. And my ongoing project is to search for the interaction of thymokinone and then other phytochemicals derived from Nigella sativa to look for this interaction and this possibility of this Nigella sativa compound to block the initial binding of the virus to the plasma membrane of the host cells. And the last parameter that I want to speak about a little bit on the next slide is the high therapeutic index of thymokinone, because when we are speaking about phytotherapy, we have the good thing and the less good things, the bad things. And sometimes you have an interesting antiviral property or an interesting biological property, but you may also encounter some undesirable side effects. And what you can see on this slide is that thymokinone is not only the most active ingredient of Nigella sativa, which block the infection of many viruses and ahead of viruses, bacteria, parasites, etc., but also it is the most safe of these phytochemicals because it is highly non-toxic. And so I think that this way of interpreting Nigella sativa and the phytochemicals and the antiviral properties opens a new route to understand very precisely how the phytochemicals from Nigella sativa works. And this is the end of my talk. I want to thank you for inviting me to this really, really interesting conference. And if you have any question, I will be happy to answer.